So, dear students, Namaskar. I'm Dr. Nutan Prakash Vishwakarma, and this is the second part of sequence alignment. So, if you have not seen the sequence alignment part 1, please go back in the playlist, watch the complete video of sequence alignment part 1, and then you can continue this particular part 2. So, in part 1, we have talked about what sequence alignment is all about, how many type of sequence alignment is there. Then we have discussed some concept between the sequence similarity and identity and all. So today in this lecture, we are going to discuss in detail about the sequence alignment. So let's recap for 1 or 2 minutes what pairwise sequence alignment is all about, what multiple sequence alignment is. So in the pairwise sequence alignment, yesterday I discussed that whenever you want to align two sequences, that is simply known as pairwise sequence alignment. And for any analysis, if you want to align more than two sequences together, that is simply known as multiple sequence alignment. So here you can see in the pairwise sequence alignment, suppose you have the sequence number 1 and sequence number 2, and after alignment you got the position number 1 in the sequence 1, there is the A. At the same position in the sequence number 2, there is also A. So simply this is match in the DI sequence. So both the bases are matching. However, at the second position you see the sequence number 1 has C, however the sequence number 2 have T. So simply it is known as mismatch. So there is two possibility, either match may be or mismatch may be. At the position number 3 and 4 again you can find the match. But at the position number 5 you see that G is there, but in the sequence number 2 there is gap, there is nothing. So what you can say? Either you can say if suppose this sequence number 1 is the older sequence in the evolution, the ancestral sequence, so in that case you can see that during the course of evolution there is the deletion at the position number 5. That's why consequently we got the sequence number 2. So there is the possibility, there is the gap, all right. But here the deletion, the event deletion. However, if I say the sequence number 2 is ancestral sequence, so how I can explain this alignment? I can explain this alignment that during the evolution there is the insertion at the position number 5. So that's why I got the sequence number 1. But just only watching the sequence number 1 and sequence number 2, no one can say which sequence is the ancestral, which one is the descendant, which one is the older one, which one is the latest one, right? No one can say. So in this particular condition, whenever you find the gap, in that case, no one can say. So that's why whether insertion actually takes place or deletion takes place, so combinedly insertion plus deletion, we are going to call it a indel, INDEL. So whenever during the alignment got such type of gaps, combinedly we can say it is indel, all right. So in the multiple sequence alignment, you find whenever you try to align all the sequences in a given query set, so this is simply known as multiple sequence alignment. Then yesterday I talked about the sequence similarity and identity. So sequence identity, how to calculate the sequence identity? See, here I have taken the example of nucleotide, all right. So nucleotide basis. So here sequence identity means exactly the same amino acid or nucleotide in the same position, for example. See, here I have given one sequence alignment between the sequence number 1, 2, and 3. Here you can see if I suppose I have done the alignment. So if I want to know that how much percentage sequence number 1 and 2 are sharing the identity, what is the percentage identity which sequence number 1 and 2 have? So how I can calculate it? Simply, if I want to calculate the identity between sequence number 1 and sequence number 2, and suppose this one I got after the alignment, all right. So just focus on sequence number 1 and 2. So the formula to calculate the identity between these two sequences is very simple. First, you have to find out that how many actual identity is there both the sequences are sharing. So here you can say you can manually match it out. All the 5 here you can see exactly 5 bases are identical, right? So what I can how I can calculate the percentage identity? I can say 5 identical nucleotides, all right, and then divided by minimum length of 1 and 2. What does it mean? Means you have to check it out that the sequence number 1 have and sequence number 2 have how many bases. So you have to consider though that sequence which have the minimum number of bases. So here in this case you can see that sequence number 2 have minimum number of bases, that is 5 only. So I am not going to write 7, right? I can leave the sequence number 1. So for the sequence number, the sequence which have the minimum basis. So in this case, the sequence number 2 have the minimum basis, that is 5. So 5 divided by 5 multiplied by 100. So you get the 100% identity. That's how. This is the way you can calculate the sequence identity. So again, if you want to check about the sequence identity between the 2 and 3. So again, between the 2 and 3, the same scenario is that only 5 identical nucleotides are there between sequence number 2 and 3, and the minimum length of the sequence having sequence number 2. So and sequence number 2 have 5 bases. So 5 divided by 5 multiplied by 100 equal to 100% identity. Both sequence number 2 and 3 sharing. And if you want to check the sequence identity between the sequence number 1 and sequence number 3, so you can compare. There is the 6 identical sequences. Here I think at this particular position you can see that this position the sequence number 1 have T but sequence number 3 have A. So 6 identical nucleotides are there, divided by, you can see the total length of sequence number 1 is 7 and sequence number 3 also have the basis 7. So simply I put 6 divided by 7 multiplied by 100. So it comes around 85%. So this is the way you can calculate the sequence identity between the bases, two sequences. Yesterday I talked about the sequence similarity while you were dealing with the, once I say that the percentage identity or similarity between the nucleotide bases, both have the same meaning. But once it is the matter of the amino acid, there is a different meaning of each of the bases, each of the amino acid sequences. Substitution with similar chemical properties. Suppose glycine is replaced by, in one sequence of the glycine at the some position, the sequence number 2 have the alanine. So you can say this is not mismatch. This is actually the glycine is replaced by alanine. So you simply this is the sequence similarity. All right. Although it is mismatch, but if glycine is replaced by alanine, it is not going to much affect the overall protein's structure and function. So GAVLI, if G is replaced by A or V or L or I, you can say this is the sequence similarity. Alright. If phenylalanine is replaced by tyrosine or tryptophan, this is again comes under the similarity. So similarity is a direct result of observation from the sequence alignment. So sequence similarity can be quantified using percentage. Alright. The types of alignment, there is two types of sequence alignment. The number one, global alignment. Another one is the previously I just categorized the alignment based on the number of sequences at a time you are aligning, right? But here my perception is changed. Now, how much area, how much sequences, how much bases you are covering in your alignment, based on that, the types of alignment is of two types. The global alignment. Number one, global alignment. Global alignment means that cover the full length of gene or protein sequence for aligning closely related sequences that is similar over their whole length. Means if you have two gene sequences or two protein sequences, what you have to do, you have to align both the sequences from starting to end. You don't have to leave a single basis, right? Means you have to include all the bases in your alignment. You don't have to leave anyone, right? Just you see there is the two amino acid sequences or two protein sequences, and both the sequences are aligned from starting to end. So when we use now the question is when we use the global alignment. So global alignment we use only when both the protein sequences or amino acid sequences have either exact length or almost similar length. If there is a huge difference between, suppose the sequence number 1 is having 50 amino acid and sequence number 2 having 500 amino acid length, in that case, if you go for the global alignment, later we will come to know that the end of the global alignment, the length of both the sequences are going to be same because gaps is going to be inserted at the end of the global alignment. So that will not make any sense, because if you have done the alignment, there is the 10 identity and then 100 gaps, then certain similarity, you will not be able to conclude anything. So now the just remember, whenever there is the similar length of both the protein sequences and nucleotide sequences are almost same length, only in that case we use the global alignment. And obviously in the introductory lectures of bioinformatics, I told you that the algorithm means the simple instruction, is step by step instruction to solve any particular problems. In computer science or bioinformatics is known as algorithm. So whatever alignment we are going to make, that is not simply possible with the help of pen and paper. So obviously we need the softwares to align. So softwares is based on certain algorithm, because once you have the algorithm, you have the framework how you can solve this particular problem. With the help of the programming language, you can implement that instruction in the form of the softwares or programs. So that is simply the logic. Whatever the logic to solve the global alignment problem is simply known as global alignment algorithm, and the example of that global alignment algorithm is Needleman-Wunsch algorithm that we are going to see in very detail in a couple of minutes. So this is the global alignment. If you talk about the local alignment is very simple, and alignment only only cover a certain region, not whole sequence. That is, for example, that is domain of the protein. I think you know about the domain is the structural and functional unit of any particular protein, of a gene of protein sequences. Local alignment is to try to find the region with highest density of matches. The tool for local alignment is based on Smith-Waterman algorithm. So whenever there is a huge difference between two sequences in which you are interested to make the alignment, so in that case you just simply go for the local alignment. It's not necessary that you go for, and although it is not required that you go for aligning of starting from base number 1 up to the end. All right. So local alignment is just simply we are trying to find out the highest density similar regions, and local alignment problem can be best solved by Smith-Waterman algorithm that we are also going to discuss in detail. So whatever the methods in this particular lecture, I am going to discuss about only pairwise sequence alignment. Regarding the multiple sequence alignment and all algorithms, we'll see in the later videos. So there is three methods for the pairwise sequence alignment. The number one is dot matrix analysis. Number two is the dynamic programming algorithm, and number three is word or k-tuple method. It may seem that all the things are very new for you. So let's discuss one by one in very detail so that everything is going to be clear to you. So first discuss about the dot matrix analysis. What dot matrix is all about. So dot matrix analysis or dot matrix algorithm is the method for comparing two sequences to look for possible alignments, right? So this was the first method that was developed by Gibbs and McIntyre in 1970. The algorithm for a dot matrix. What we do actually in the dot matrix analysis or in the dot matrix algorithm, what we do actually, one sequence is listed across the top of the matrix, and the other, which you can call as a sequence number B, is listed down the left side. All right. So starting from the let me do it, then we'll come for the explanation of it. What we do actually, see here I'm explaining you all the written material you can read for your examination point of view or for your understanding point of view. So once I read each and everything, it will be confusing for you. So what I do is simply I'm explaining here, then you can go for the reading of the text, whatever I have written. So suppose you have two sequences. This was the in 1970 that method was developed by Gibbs and McIntyre. So this is very primitive, and currently we don't use it. But if we modify in this one, the things is going to be better, right? So this is the very primitive. That's why I am explaining you so that you can understand the concept behind the sequence alignment. Just if you know the history of the bioinformatics, in that one I told you that first whenever the protein sequencing was done, and that was very common among the scientific community, so many labs were started making the uh, databases, so that the sequences, the informations can be spread throughout the globe, and any person sitting over in any country, he or she can retrieve the updated protein sequences. So there is the race. And later you know all the history that the first database was created, the Atlas of Protein Sequence and Databases. Then it was that is the era of flourishing of databases. So there are the very good databases was created. Now scientists were started realizing that if we only spread the databases, only the information, until we don't analyze them, we will not be able to reach up to any conclusion. So obvious question is, whatever sequencing have been done in the laboratories, is there any similarity between the two protein sequences? So now the $1 million question scientists would have at that particular time, that how to align these sequences, whether they are sharing the similarity or not. So simply Gibbs and McIntyre comes for the rescue, and they develop one method, that how to align actually the two protein sequences or two nucleotide sequences. So what Gibbs and McIntyre says, that one sequence, obviously you are going to make the pairwise alignment, so only two protein sequences align you have. Here in this case I have taken the example of the nucleotide, you can take the example of the protein sequence. But for the ease of doing, I am putting here the gene sequences. Alright. So first sequence I have write down horizontally, and second means on the top, and second sequence I have written over the left side. That you have to do. Now what you have to do, you just start from this, and then suppose just how many bases are there in the horizontal one, this much column you should draw in the table, and how many bases are in the left side, that much rows you have to draw in this particular table. All right. So now what you have to do, you just start from moving from right side direction. So if I move from this direction, you can see the cursor. So here you can see in the top and in the left, here is the A and here is the A. In both the cases there is there. So whenever there is the match, what you can do, you can plot a dot. See, I can plot a dot. Then I can move ahead horizontally. Then here I can say here is the A and here is the T. So obviously you don't have to do anything. Just keep moving. Here is the A and top it is G, no matches. Then AT, AT, AC. So there is no matches at all. Next, now I can start from this one. So here is the T, here is the A. So you don't have to do anything wherever there is the mismatch. If I go from this T and here is the again T, so I can get a match. So what I can do, I simply can put a dot. Then you can move TG, TT, yes, TT. Again I put at the dot. TC, there is no match, so I don't have to do anything. What about the G? GA, no match. GT, no match. GG, yes, match is there. And you don't find any matches here. If I start from the T, I can get a dot. Here I can also put a dot. Here I also can put a dot, and here is no match. Again for the T, again for the T, and again here is no match. From the C, I can get only one dot. Yeah. So these are the, simply you are putting the dot wherever you are finding the matches. So that's it, and you can find a beautiful matrix. That's why this is known as dot matrix. How to interpret what this matrix conclude? Okay. So if you see the diagonal line, here you can see the diagonal line starting from this point to this point. You see, you can find the straight diagonal line. So this is the simple diagrammatic representation, that if two sequences are showing the similarity or identity, here in this case obviously because I'm dealing with the nucleotide sequences, there is no meaning of identity and similarity, because sequence identity and similarity are both the same thing in the case of the nucleotide sequences. So if two sequences are identical, in that case you will find the straight diagonal. So this is qualitative estimation, that whether two sequences are identical or not. Here just for the sake of demonstration, I put it the very small the bases, very few bases. But if it is the matter of the comparing of two genes, suppose globin gene of human and globin gene of mouse, so the length of both the genes are very much, it's too much. So obviously with the help of pen and paper, we will not be able to make it. What we can do, we can make this, there is certain software. This logic is implemented in the form of the softwares, and once you put both the sequences in that particular software, you will get the automatically the dots. You don't have to do it with the help of pen and paper. So let me show you some screenshots of software. What can dot matrix do? So it can detect matching regions. It can be used to assess repetitiveness in a single sequence, such as direct and inverted repeat within the sequences. See here I've taken the one software is available. I will demonstrate it in the practical session. I will upload a separate video which will be based on the dot matrix analysis. So here is the screenshot I am sharing. So suppose two identical sequences we have put in. So in this case, you can see you can find the straightforward diagonal line. So you can conclude that both the sequences are identical because you can see the continuous identical, the no the diagonal line, and these all the small dots are also showing the similarity somewhere at the particular basis. By using certain methods, you see whenever the bases of these two sequences are going to be large, so number of dots is going to be increased, and figure is going to be image is going to be really dense. So what you can do, certain improvements are there in the DNA dot matrix. The sliding window approach and certain thresholds are there that I will explain in the practical session. You can reduce the number of dots, okay, so that it can be easily interpreted by the researcher whether two protein sequences or two DNA sequences are sharing the similarity or not. So here in this particular case, you can see you find the continuous, the straightforward diagonal line. So you can say that both the protein sequences are what it's identical sequences. Here I'm sharing the second one. Here you see, if I put one sequence, two sequences for the dot matrix analysis, here I find just only dots. This is called noise. Here I am not finding any diagonal line, straight diagonal line. So you can say that both the two sequences were very different. All right. So that's why the qualitative estimation is here is there. If I see the third one, you see here I am finding the diagonal line, but here you see, if you see very closely, you can see that this diagonal is not straight. No, there is the interrupted. Here is the gaps, right? It's not continuous. So you can say here is again it's not continuous line. So here you can say that two protein sequences are similar, they're not identical, somewhat similarity they are showing. So what is the limitation of that dot matrix? This dot matrix gives you only qualitative estimation. Suppose, let me give you one example. Suppose you have one gene, the globin gene, and you have the protein sequence, globin, and you have the gene sequences of 10 organisms at that particular time in 1970. If you want to know that human gene is showing maximum level of similarity with which particular organism, so what you can do? First, you go for dot matrix analysis of your sequence with the organism number 1, organism number 2, organism number 3, and organism up to organism number 10. Then you go for the just analysis that in which particular alignment you are getting the longer diagonal line and straight diagonal line. So the sequence, the matrix which gives you the longer diagonal, you can simply conclude that your particular sequence is matching with the sequence of this particular organism. So that is the only qualitative estimation. The limitation is you will not be able to judge that if there is the minor differences in both the alignments, you will not be able to judge that which organism is actually showing the maximum level of the similarity. So problems with the dot matrix method was requires human supervision, large memory and processor time it needs, right? No explicit handling of gaps, as already discussed about in the pairwise alignment. I told you there is the indel, like insertion may take place or no deletion may take place. So there is no explicit handling of the gaps. So here no concept of the mutation and all. and no good quantitative score of alignment quality, we will get only and only qualitative estimation whether two protein sequences are showing their similarity or not. So that was the limitation. So now we are going to discuss about the new algorithm which is better than this previous one. So that we are going to, for that one, I'm going to upload a separate video. I hope the concept is very clear to you. You learned about the dot matrix analysis. So in next lecture, we'll discuss about the next algorithm. Till then, take care. Have a nice day.